Got evidence for evolution? Bring it. Let's see it. I've put the link to this stream in the chat and in the description of this video. So if you want to join and share evidence for or against evolution, I invite you to come and do that. You don't have to have any scientific credentials. If you have a piece of evidence that you want to share, you can come and share that. And if you do have scientific credentials, great. I also welcome you. So I'll just start by sharing some evidence against evolution. And if anyone joins, then we'll go from there. So I was shocked yesterday to find this paper, as this is in a peer-reviewed journal, and I was absolutely shocked by the title. It says, "Biological evolution is dead in the water of Darwin's warm little pond." I mean, this is unbelievable. I believe this is so true, but just to see this in a peer-reviewed journal in academic circles where evolution is assumed to be true, it was pretty incredible to see this. Now, the specific problem that's being referenced in this paper and why they say that biological evolution is dead in the water of Darwin's warm little pond is because they are picking out one specific problem, which is the co-origination of enzymes and vitamins. And they're talking about how, look, natural selection really, it doesn't have the power to facilitate things that have dependencies on each other in order for origination. Meaning, if you have an enzyme, which, if you're unfamiliar with that term, it's really a machine inside of our cells. And we have many enzymes in our cells that do fantastic work without which we cannot live. And so for any one enzyme to originate by natural processes is already absurdly improbable. But this paper is specifically talking about the problem of, even if you got one enzyme to come about by natural processes, then it's still not going to be selected, because a lot of these enzymes require other enzymes, and they require vitamins and all kinds of things that have to be present at once. And so this makes the really improbability of one enzyme coming about by natural processes into a much bigger, absurdly implausible scenario of enzymes coming about at the same time when they are needed to support each other for life. So that's what this article is about, and I encourage people to read this if you want to know more. I'm just going to read a highlight from it. And so it says, we propose, okay, now this is what I already told you, co-origination. I summarized this for you. Now, I saw this in the conclusion, and it's just hilarious. I can't believe this got through peer review, and I'm going to talk about why it might have got through peer review here pretty soon. But it says, science has spent more than a century and a half worshiping at the altar of an idea that has few scientific clothes, surely no ensemble. We are on a yellow brick road leading only to a wonderful wizard. Like Alice, we are not in Kansas anymore. Natural selection is not natural, but is given powers that are magical and miraculous. Woohoo! I'm so glad someone was able to say this and get it into a journal, because this is just expresses my feelings about it so beautifully. But the question that came up for me is, how did this get into a peer-reviewed journal? And I'm not saying that because of the science it's based on. I think the science is solid. what I'm saying is, how did it get in when we have such a stronghold in academia that favors evolution and assumes evolution to be true and dismisses anything that doesn't adhere to that sacred cow? Well, it's pretty interesting. I read this blog post from, let's see, what was his name? Lawrence Moran. And he talks about how did this get through peer review. And he mentions that Dennis Noble was one of the editors of this journal. Now, so he says we can assume that Dennis Noble is responsible in part for allowing these papers to be published. So Dennis Noble, for those who are not aware, and he believes in evolution. He's a scientist who believes in evolution, but he recognizes that mutation plus natural selection is not cutting it. And so he's basically saying we need to throw out this idea of this is how evolution occurs. He still believes in evolution. So he's promoting other ways, other mechanisms for evolution. Now, I disagree. I don't disagree with the mechanisms he's proposing, but I do disagree with the conclusion that therefore evolution can happen. Because I think the problem with the mechanisms that he's proposing is that they cannot get genes to originate. You still have, and there's many other problems too. You still have all the problems trying to get life to form by natural processes. But once, let's just say you have the first life, we'll skip all the other problems and we'll go straight to trying to get life to diversify. You still need a gene originator. There's a lot of mechanisms that can move genes around, right? And develop different things that are already present in the genome of the organism. But where do those genes come from? We've got to get genes for all the creatures and plants and everything alive on Earth today. And we don't have a mechanism that can make those genes originate. So that's what I have to say. And now I want to see if anybody wants to join and share your thoughts, share your evidence for or against evolution. I'll just read some comments until anybody happens to join. And if nobody joins, then I'll just shut it down. So Grayson, good to see you. And I'm ready to hear, we can unpause the conversation if you want to finish what you were talking about the other night. He says abiogenesis and evolution do not say that enzymes just pop into existence. LOL, this is a straw man, right? But there has to be an accounting for how they came into existence. And so trying to explain how they come about by natural processes, well, we don't have scientific observations to show how they could come about by natural processes. So that's the problem. I'm not saying they just pop into existence. I'm saying getting them to get there, however long you have, by natural processes is not something that is, it's absurdly infeasible. So, okay, Michael says you can't debunk evolution by debunking abiogenesis. Well, what's interesting about that is that there has been a real push to separate evolution and abiogenesis. And of course, people who believe that things, that want to promote evolution want to separate themselves from abiogenesis because they know it's a losing cause. They know they've lost that. So they want to separate themselves, get them away from that. So they want to push off all the hard problems to abiogenesis. But then I noticed that the abiogenesis people, they want to push off the hard problems to evolution. So abiogenesis people will say, but the first living cell probably didn't have that. The first living cell probably didn't have that. So they're trying to push the hard problems back to evolution. Well, let's just put it this way. We are talking about the origin and evolution of life by natural processes. They go together. Okay, so this is a philosophy that things came about by natural processes. That's a philosophical framework. And it's one that I reject. And I reject it on the basis of our scientific observations. So for the purposes of arguing with people who want to supply evidence for evolution, I'm happy to give you the first cell, but I do not think that it is legitimate to try to completely separate abiogenesis from evolution. It's just a ploy to help your cause because you guys want to just push off the hard things to the other side. So, and Michael, thank you. Yes, he said the reason why it got through peer review is because this is Dennis Noble's journal. Thank you. And Grayson said, so last week, Rebecca was confronted with unassailable evidence against her position, but rather than change her mind, we see her having a clear emotional response to confirmation bias. Well, actually, I'm still in ongoing talks about our discussion last week, and I hope Dr. Dan and I can continue that discussion, or I'm open to discussing that same point with anybody. And what Grayson is referring to is the demonstration that Dr. Dan gave about common ancestry versus separate ancestry. So the jury is still out on that one, and I'm waiting to have issues resolved. And I have to be honest, I would love to give a few points to evolution. What I mean is right now, I feel like everything is stacked on my side. So I would be happy to at least like give some crumbs to evolution. So I want to be convinced of this. Okay. And Grayson, I welcome you to share, finish sharing what you were sharing the other night, because I know we didn't finish that conversation. So, but right now I can't credit you with giving me evidence for evolution, but I do want to be able to credit people with giving evidence for evolution. Okay. So, smiling and cheering to anything that conserves or confirms her preconceived notion. Yeah, come on, it's just having a good time here. Raised by wolves, that's good. She's keeping her mind open. We all should. Okay, Michael says, not open enough to actually make an honest attempt at understanding why basically every single biologist accepts evolution. Well, I'm attempting to understand, but I have a feeling, well, I just leave it open. I'm open to people sharing their evidence, and I'm considering the evidence, but I don't just go on, I don't make my decisions about what to believe based on numbers of people that believe them. And so I have a problem with farming out my critical thinking to other people and to the majority, and especially on something as significant as this. Jamie says abiogenesis has been demonstrated. We don't have to debunk it. Oh, I think he means we don't have to debunk it. It isn't established. Yeah, we just point out how you need what you don't have. Yeah, has not been demonstrated. Correction. Thank you. Grayson said, Rebecca, even if God created the first population of cells and naturalistic abiogenesis is false, evolution could still be completely true. Absolutely right. Yes, absolutely right. If God created, he could have created by a miracle, created the first life by a miracle, and then it could have developed by natural processes. But if I'm to believe that, I would need to see a lot of evidence. Right now, I don't have a good reason to believe that because the evidence is strongly against evolution. Now, I know that people will balk when I say that, because what I think evolution is strong in is rhetoric, and the numbers of people who believe evolution, the numbers of people who completely accept this as true, and the stronghold in academia and in the media, that is the strength of evolution. When I actually look at the evidence, I don't see the evidence for evolution. So I think evolution's strong point is in the stronghold they have in the media and in academia. But hopefully that will change. And so I would like, and if I'm wrong, then please show me how. That's what this stream is about. I'm giving people who believe in evolution an opportunity to show the evidence. So Jamie says, don't give it to them, Rebecca, they don't deserve it. Okay, I think he means giving the points. Yes. So if one can be true while the other is false, that means they're separate. Yeah, but let's not be ridiculous here. I'm talking about, yes, absolutely. But we could separate every point of evolution. We could say, well, not only did God do a miracle at abiogenesis, at the first living cell, but then he did a miracle halfway through evolution. And he did, and evolution, I think it does need miracles, honestly. So I think that's the only way that evolution is happening, is with a whole lot of miracles. But to say that that means they're separate, I don't think that's true, because what we're talking about is the idea, the philosophy, the understanding, the worldview that says we came about by natural processes. And that is the worldview that I think actually contradicts our scientific observations of the world. When we observe the things that are around us, plants, animals, ourselves, consciousness, everything, these things point to an intelligent designer. We know what something looks like when it's designed. We have lots of examples of designed things. My iPhone is clearly designed. Well, the systems in our body, the integrated systems in our bodies, are vastly more complex than my iPhone. So we can look at the iPhone and say that definitely had to have a creator. And so yes, we needed a creator. Our bodies and all the living systems that we see on Earth show clear evidence of intelligent design. That is where our scientific observations lead us, if we are not trapped into a framework that forces us into thinking according to natural processes only. So Raised by Wolves says, I mostly accept evolution, but I don't have my day ruined because someone somewhere doesn't immediately accept every single aspect of the theory. Cool. Thank you. I appreciate that. Evidence and Reason, Sal says, even Dr. Dan admits there's no universal common ancestor for all major protein families. Yes, that's true. And Sal, you never get tired of repeating that, do you? Sal says, common design can also make nested hierarchies. Common design is a better explanation for supposed co-evolving nested hierarchies, such as binding interactions and nuclear localization transport. Sal, why don't you come on and share about that? So I'd like a bit better explanation on the nuclear localization transport. Grayson says, common design cannot sustain nested hierarchies. Mutations would break them. Now, okay, look, nested hierarchies. We've been talking about that a lot, and I'm going to separate this from my Dr. Dan talks because I want to complete that talk with him separately. But I will tell you, there are so many problems with nested hierarchies. Oh, where do I begin? First of all, we have to decide how we're going to group animals together. Are we going to group them by morphology, or in recent times, we're trying to group them together by genetics? But what happens when we do this? We have problems because we have some organisms and the proteins is also a problem. If you try to organize them by which proteins they have, that will give you a different tree. So if we all descended from a common ancestor, then you would expect that everything would match up, right? Like morphology would match with the genetics. It would match with the types of proteins that are in each organism, but that's not what we see. And what I've noticed is that the trees, when they get broken, like basically when the tree doesn't work, evolutionists invent words to try to make it okay. Oh, it's all okay. We have convergent evolution. So convergent evolution is just a word that's made up to patch up a major problem with the tree of life. And that is, we find organisms with very similar morphologies that are not related and not on the same branch of the tree. They're like branching way out. How did they end up with these similar features or with similar genes when they're so disparate? So, okay, Sal is joining. Good to see you, Sal.
Oh my goodness. I'm sorry, I had the YouTube playing at the same time. How can I be of service?
Well, why don't you tell us about what you were talking about with the nuclear localization transport?
Yes. First off, there is a divide in the creation and ID community whether nested hierarchies exist. I'm emphatic that they do exist, but there are problems with it because, one, as you know, some of the nested hierarchies don't exactly agree with each other. But I've seen it in my own work, and I published with Joe. By the way, Joe DeWeese, just so you know who I am, I was a co-author with Joe DeWeese at Oxford University Press. He recently published a major secular peer-reviewed book. He was the editor, Springer Nature. So that's a magnum opus. He's a respected expert, and he's a creationist. So how that relates to the nested hierarchies, some of our publication kind of depended on it, honestly. But going back to your question about nuclear localization and transport, it's like when you change a key. Like, let's say you have a master key that opens all the locks in a building. If you change that key, you're going to have to change all the locks as well. So when you have like a protein that interacts with like 100 other proteins, like say in a plant, and then you do the same for a mammal, what the evolutionists don't do is they don't account for the fact that if you change something that interacts with 100 other pieces, like a master key lock, versus all the locks, you're going to have to change all 100 simultaneously, especially if it causes death. That's pretty bad, and you don't change it. So nuclear localization, that's kind of a fancy word, but just think lock and key.
Okay, but what is the key? Like, where is the key? Like, can you explain what is the process in the cell? Like, what is this thing that you need a key for?
Okay, so the first thing is for eukaryotic organisms, what defines the eukaryote versus a prokaryote is it has a nucleus where a lot of the DNA transcription and RNA generation happens. And then also, for that to happen, you have to have proteins go into the nucleus. And they need a special password to get through. That's called a nuclear localization signal. And if you change the gatekeeper, which called it an importin, you're going to have to change all the proteins because all those proteins have in the protein itself, it has a code. It can be anywhere from 4 to, I've seen up to 24 characters, and they correspond to English alphabetic characters. This is like a password. So if you change the importin, you're going to have to change like all 400 proteins simultaneously. And so you're going to have to change you basically have to co-evolve all the nested hierarchies in a coordinated way so that the creature doesn't die. But this isn't just true of nuclear localization. Anytime you have a protein that has to interact, and there's a chain of 100 proteins in a process, they have all these, basically the way they connect, it's the kind of an archaic metaphor is lock and key. So you're going to have to change all the locks and keys so that these things can join. They can find their soulmate, so to speak. And they never account for that. When I was talking at a conference of professors who are ID friendly, they're biochemists, cellular biologists, people in the pharmaceutical industry, medical doctors. When I pointed out all the coevolution that you would have to have, we all, and then the evolutionists say, we made a great discovery, all these things co-evolve. We all laughed. We laughed. And I told Dr. Dan, I said, you don't realize how much we laugh at you guys because you just make these assertions and it's like you don't even think about it. And he said, Sal, that's actually a good point. Maybe you should write that up. I'd be curious to see what the responses are. I'm like, yeah, you should be curious. You should be curious even before I write it up, why these aren't problems. So we see that with the zinc finger interactions that are transcription factors. It has to find, okay, so you have the DNA. It's spread in the human body. It's like what, 3.3 gigabases. How do we know to activate a gene at the right time? You have to send the machinery to the right address. So if you change the address, you also have to change you have to change the address, the physical location, but then you have to update the postal delivery guy, which is the transcription factor machinery, and you can't.
Are you saying there's currently no attempt at an evolutionary explanation for that?
There's no attempt, except they'll write peer-reviewed papers and they say, we discovered this coevolution. And I'm like, we just laugh. I said, well, like, as if you discovered like the origin of life. It's like, yeah, we're alive, so life originated, but you don't have any mechanistic explanation why this should happen. And I've confronted, let me see if I can get this a little better. I've confronted evolutionary biologists about the problem. Half of them don't understand the mechanics of protein interactions because they're all stuck in phylogenetic reasoning. It's like, you guys don't work with proteins and binding interactions and the difficulty of forming them. If you did, you'd be a lot more reserved. And the other ones just say, well, what a great discovery. Evolution works better than we ever imagined. And we just laugh. It's like, yeah, let's admit that it evolved, but it's like, isn't that a miracle you need? And they won't admit that. So,
yeah,
I confronted Dr. Dan on that. He said, that's a really good observation. I think you should write that up. And I'd like to see what my colleagues think. And that's actually how come I ended up speaking at Evolution 2025. It was because he was curious. And I said, well, I'm not going to cover that topic. I covered another topic instead. So I just wanted to say, a lot of ID proponents and creationists, myself included, believe in nested hierarchies. But we believe they're designed, and we're actually seeing evidence that it's extremely well designed, so much so that we can run what we call structural bioinformatics and predict protein structure, which is kind of voodoo just by looking at the different. Okay, so you have the shape of you've seen.
Wait, hold on. I want to talk more about that. I want to get into understanding what you're saying about the nested hierarchies, but I want to address this comment from Dave. He says, even if the criticisms of current evolutionary theory were correct, why would we posit an imagined supernatural cause for complexity rather than an unknown natural cause? Well, it's up to you what you want to do with it. So I'm not trying to take anybody's natural processes away from them. If that's the way that you want to live, the reason I'm doing this, and I'm admitting my agenda, guys, I have an agenda for this whole channel. My agenda is that people would know the truth about reality with regard to our origins. And the truth that I want them to know is that the scientific evidence shows that it's a strong indicator that we were intelligently designed, that there is a creator. And so I want to give that gift to people who have been blinded and who've been taught that everything comes about by natural processes. And that is just simply not a scientific view. It's a belief you can hold, but it doesn't line up with our scientific observations of the world. So that's my agenda. I want to help people who would like to believe in a creator, but have been taught falsely that evolution and abiogenesis, that there's tons of evidence for it. So we have Grayson is joining. How you doing, Grayson?
Hey, Rebecca, long time no see.
Hey, it's been like a week.
Yeah, I wanted to ask Sal if he was aware of Dr. Dan's model that he was showing you last week about how nested hierarchies that we observe are not possible under common design.
I saw part of it. It can be possible. I mean, a creator can do anything. So, sorry.
But okay, Dan's argument was that even if the creator created a perfect nested hierarchy at the start, it's the accumulation of mutations post-flood, post-fall, that would break that nested hierarchy.
A creator can do anything.
Okay,
But you
Grayson,
agree
let
though
me address
that these
this.
mutations, okay, if you're
No,
accumulating random mutations, that
no,
there
no,
would
look,
not you would not
look,
expect these mutations to be in the same places if they're accumulating independently along separate lineages that were not sharing a common ancestor.
we don't know the mutation rates. This is to extrapolate what we observe now and then extrapolate in the past. And fundamentally, a creator can do anything. He can
But
accelerate.
regardless,
A creator can do anything. He can accelerate. So I'm sorry.
no matter what the mutation.
Can you mute your mic when you're not the one talking? Because there's a lot of feedback coming. So,
Oh, really?
Yeah, it's very noisy on my end. Probably, yeah.
Okay. Yeah, I'm saying regardless of the mutation rate, it doesn't matter what the mutation rate is. It's the fact that the mutations are shared in this same loci across branches that should be totally independent under common design.
I have a creator can do anything. I don't know how else to repeat it. I will point out, I myself have seen in the lambda interferon 3/4 the same mutations in the pseudogenes of chimps and humans. That has been an argument for common descent.
Mm-hmm. Yeah.
But also it has said, okay, so this caused damage to both lineages. We have immune disorders in both lineages. Now, this could also be a miracle of God's judgment, which is consistent with the Bible. But, the thing is, you're straining at gnats. You have much bigger things to deal with nested hierarchies than straining at this gnat. And I get after creationists. I said, you're letting people like Dr. Dan define what the real argument is. The real argument should be, how do you have all these coordinated changes? If you're going to strain at those little gnats, you're letting camels through, and those are the nested hierarchies that have to co-evolve. I can't tell you, this is unbelievable. You're not even dealing with nuclear localization signals. You pretend it's not a problem. You don't deal with binding interactions. You don't deal with transcription factors having to be tuned to the DNA promoter regions when they evolve. You're straining at gnats, letting camels through, and a creator can do anything. And what Dr. Dan is doing is defining what a creator ought to do. That's not right. And that's fine. Look, I actually did credit Dr. Dan that he made a very good analysis, but I've always complained to him, you're straining at gnats, letting camels through. There are big elephants in the room. You pretend they don't even exist. You just deal with this little gnat. You pretend like
I think I appreciate you.
You're letting all these huge elephants in the room get by. I do get on the case of some creationists. I say, look, you may have a point there, but we have like a sledgehammer point that you're not even addressing. I have a problem with that.
To be honest, I think that this point is a sledgehammer point, and I appreciate you trying to pivot to other subjects, but I just want to make it clear here that when you're saying that God can do anything, the thing that you're claiming that God is doing is mutating separate lineages of independent, different kinds under common design, and he's giving them mutations to make it look like they share a common ancestor. That would be deceitful.
Okay, wait. Sal, you have to let me jump in here because I think Sal gives too much credit to the evolution side. So I know you think it's gnats at Sal and that we should focus on the camels, but I want to focus on this gnat because I see some major problems with what Dr. Dan was doing. And so first, Grayson, do you recognize that like what we're talking about, you guys keep switching from like talking about how we have to get so many variations from Adam and Eve, right? And so, but remember, the demonstration was not about how many variations we have to get within humanity. If you guys want to have that in a separate talk, we can talk about that. And if there's problems with related to that, then related to the variations that come up within humans and the number that we need to get, then we can talk about that separately. But remember, the demonstration is specifically humans to chimps to gorillas, right? It's a comparison between three. And my problem with it remains the same. You guys don't have a scientific starting point for which to make that assessment. You're saying that, Dr. Dan gave it favorable conditions for the nested hierarchies, but I'm going to ask again the same question that I asked before. You guys said I could adjust the parameters, and I asked you, why can't I adjust the parameters to have the 5 bases that you used on the common ancestry? Why can't I have those 5 bases for separate ancestry?
Well, Rebecca, do you remember what my response to you was last week?
Yes. And I thought about it, but I realized you were wrong.
The starting for common ancestry is that they all start with the identical sequence because they start as the same population. So you don't start out with 3 separate groups. You start out with one group that then evolves and differentiates into 3 different groups that inherit the same mutations. So if you start from the same place, then you would be conceding common descent.
But what you haven't shown is why 5 bases is the same ancestry. So you're assuming that, and I'm saying no.
I'm not. Look. The only thing, I'm trying to, maybe did you rewatch the video or something? Or if you look at, you start out with a certain sequence of 15, right? So in Dan's simulation, the common descent has all the same sequence at the start. It's one population. Those 15 are the same. You cannot start out with that because then it would be the same species and you no longer have separate kinds. You can pick 5, you could pick 10, whatever the amount you're trying to mutate is, but you mutate them independently across the different kinds. That's the assumption. And Sal is saying that God can introduce mutations however God wants to, and he can make it look like they're inheriting the same mutations from a common ancestor, which is what we see.
Rebecca, can I just share one little slide?
Yeah, but is it going to stay on this topic, Sal? Because I don't want to go to another topic. Okay.
It is, because this is addressing Grayson's point about God being deceptive. So if we look, this is just a metaphorical illustration here. If you could add the little pencil, the bent pencil. I had a picture of a bent pencil. Okay, is God being deceptive because that pencil looks bent?
Well, this is
Grayson, tell me, is God being deceptive because that pencil looks bent to our eyes?
No, because the consensus in science is not that the pencil is bent. The consensus in science is that evolution happens.
It has nothing to do with consensus. It has to do with optics. It's not consensus.
No, I think that it has everything to do with consensus.
I'm just pointing out, okay, I could stop sharing this, but I was going after your point. Just because it looks like a nested hierarchy doesn't mean that God's being deceptive any more than that pencil looking bent, because a common design with covariation totally means the nested hierarchy is designed. And yet I gave you good examples why an evolution random process will not create those coordinated co-evolving nested hierarchies. You totally ignored it. You're going to strain at these little gnats, which Rebecca doesn't think exists. And so, I think this thing, it fails on that point alone, on the problem of coevolution, which you don't, it's so over your guys' heads, you don't even think it's a problem.
I just didn't respond to it because we weren't on that topic. I'm happy to address that topic later, but you already did promise Rebecca that you were not going to move to that topic, and you already did. You couldn't even get through one response without going back to it. Let's get to that later, but let's just address one topic at a time here. So I'm very happy to address that. But I want to address your broken pencil thing, okay? Because you said it doesn't have to do with consensus, but I really think it really does have to do with consensus, because the reason I would say that God is being deceitful in that is because the way that these mutations have accumulated has caused the majority of biologists and scientists to conclude that this is evidence for common descent. So it's not like that the broken pencil, it's not like optical illusions have caused the majority of people to go astray and to go astray from God's reality. That is not the case. So that would not be an example of God being deceitful, because people are not being deceived by refraction. People, in your opinion, are being deceived by the nested hierarchy patterns of mutations. So that is a deception. So if God is doing it, that makes him a deceiver.
Okay, I totally disagree with that point, but like about the God being deceptive. Okay, I don't think it's God's fault that we make up bad ideas about how things came about by natural processes, and then we like find ways to justify it, and then when it, we present something, then it like God, then we make God into a liar. But that, to me, this is kind of a side topic. I really want to stick on this topic of what's going on with that demonstration. And I understand why you're saying what you're saying, Grayson. You're saying because those 5 bases are all the same. If I chose those 5 bases, that means I'm choosing the common ancestry model. But remember, we are trying to figure out, how did we get to the variation that we see today, right? And so I'm saying, look, if you were taking the 15 bases, and basically, like, what I mean is, you don't have a justification for why you guys get 5 bases that are the same and I only get 3. Like, you don't have a justification for that.
Yeah, the justification is that they're inherited from a common ancestor. Okay, like if your great great grandma has a mutation, you inherit that mutation, Rebecca. That's why it's the same.
But there's another option. That's the thing, Grayson, is that, so remember, I'm not asking for 15 bases out of 15 that are common. We're both knowing what the variation is now. The question is, how do we get back to a starting point? And what I'm saying is, and I'll try to say this in a way that can be understood, but you're assuming the starting point for human, chimp, and gorilla. And what I'm saying is, you don't have a scientific basis for that starting point.
No, I'm not assuming any. Look, I'm saying based on combinatorics, how many possible starting points could you have for 3 groups that have 15 base pairs, right? You can look at any single combination of those 15, literally use any mathematically possible starting point, exhaust every single available option, and you will see that the option that Dan went with was the most favorable, because he gave the highest, like the most perfect form of nested hierarchy to start with.
No, I have some data points. I just realized this. The evolutionists are cherry picking certain things, because this does not happen for histones. Histones are identical practically almost all the way to yeasts. So the histone in histone 1, histone 3, histone 4 in humans is identical to chimpanzees. So you're going to take the most variable proteins and start making an argument in nested hierarchies with there. See, no, this is cherry picking on your point. There's no variation in histones, and this is
full genome, Sal, full genome. We're not just going to look at certain proteins that are highly conserved, because that would be disingenuous. We're going to look at the total amount of variation.
See, that's the thing is, so you're gonna selectively apply nested hierarchies, except when it doesn't work.
No, here's a great idea, Sal.
And you're gonna extrapolate these mutation rates when there's obviously none in the histones. And by the way, I need to point this out with the histones. Michael Behe, it's a forgotten paper. He said we have these histone deletion mutants, and it kind of falsified the idea that they're conserved because of natural selection. So you could say all you want, but this is obvious cherry picking this is obvious cherry picking on the part of evolutionists. I just realized that you're building all this case. I could point out, look, there's cytochrome. There is like a cytochrome.
Well, before you move on, Sal, you made one point.
I haven't finished my point. You have the cytochrome.
You're moving on to more points.
C that is 100% identical between chimpanzees and humans. How do you like that? So now you're going to invoke these mutation rates that aren't consistent across the whole genome.
You're making more and more points.
This would absolutely support the fact that the mutations are.
You got to wrap it up, buddy.
Listen, you need to stop interrupting me, and then I'd finish my point. The point is, you're cherry picking the variation and extrapolating it to everything. That's illegitimate.
Okay, well, I agree that
stop interrupting me.
I agree that using histones alone is cherry picking, because you're hyper focusing on an area of the genome that is highly conserved and has good functional reasons to be under natural selection. Now, if you want to actually have, like, you know, a genuine look at the state of evidence, you could use part of the genome that is not under natural selection, like, say, maybe the third wobble position in these proteins. That could be multiple different variants, and you would get the same amino acid sequence in the protein. Oh, but if you do that, it replicates the phylogenetic relationships that we see in a nested hierarchy. If you look at non-coding DNA, it replicates the same phylogenies. If you look at the total genome, not just hyper-fixating on one specific protein like you love to do, if you don't cherry pick the genome and you look at the whole thing, you get the same phylogenetic relationships. I'm not the one cherry picking, Sal, you are.
Okay, now, Grayson, I don't remember off the top of my head what was compared with the human chimp gorilla paper that we're talking about. Do you remember?
Like, in terms of, like, I think it was just total genome differences. Let me look at I'll pull up the paper though.
Okay, yeah, that would be good. Because
are you aware of this paper as well?
Sal, he's asking you, are you aware of this paper?
Oh, was he asking me? No.
Okay. So, but
I'll put it in the chat.
Because Grayson, I'll tell you while you're pulling it up, what I noticed when I read the paper is that what it seems to assume as I'm reading is that it assumes separate ancestry by natural processes, meaning what would we because it compares common ancestry versus separate ancestry. But the separate ancestry, it seems to assume, is separate ancestry by natural processes. Like, what would we expect to occur if, like, it was just random, right? Like, if like, basically they didn't start with a common ancestor. So it doesn't assume separate ancestry with a creator who may choose the same genes for the same types of
that's false. Like,
show me where in the paper. Show me where in the paper.
Yeah. This is a similar criticism that Rob Stadler had of it, and he actually just he was corrected by the author himself, let's just say. But in the paper, what you're reading about it, like, is that each kind accumulates mutations randomly by natural processes that we know about today. So that's where Sal was coming in and saying that God could give each of these kinds mutations in the pattern that we see today for some reason that he doesn't know. But the paper is assuming that they're accumulating mutations independently and randomly, like with natural mutation processes that we are aware of today. It doesn't actually assume anything about the starting states. Okay, and Dan and I tried to explain this, where his simulation starts with starting conditions for these genomes. This paper that I have linked in the private chat, if Sal wants to check it out, does not assume starting states. It is examining every possible starting state for separate ancestry models, for each species being created separate and each family being created separate, compared to a common ancestry model. And that's what it's testing.
Now this is where you guys, I need to see, where is the beef? Because I'll tell you, if you're going to test every possible scenario, I don't know that how much math that would take, because how many possible scenarios are there? It did, what I can tell, 2 scenarios, one for family and one for species. And you guys keep saying that it doesn't assume a starting condition. I understand perfectly that they're starting with what we see and they're working backwards. But what I'm asking is, if you are comparing common ancestry to separate ancestry, you must have an idea about what separate ancestry looks like. You have to have, you can call it a starting point or not a starting point, but whatever it is, you have to have an idea about what separate ancestry looks like.
Yeah.
So where are they getting the idea about what separate ancestry looks like?
Based on what most probably matches the data. So they're saying, like, if these different species originated with, like, at the family level, or were created separately at the species level, what is the most probable, like, way that they would get to the condition that we're in now? So it's trying to be as maximally favorably possible to the separate ancestry model. It's really trying to help you there. I mean, it's like, it's again going through all the possibilities.
No, but the possibilities based on what?
And I did look at the study right here, and it says that the sequence data included roughly equal amounts of coding and non-coding sites from autosomal regions of the genomes, and a few thousand sites from X and Y chromosomes. I think they used 54 nuclear genes, 191 taxa. So they're going through, like, what areas of the genomes that they're comparing.
Right. Okay. So now, how do they know? How do they know what separate ancestry looks like?
They're saying they're comparing either at the family level or at the species level. So
How do
what
they
are the
know what
odds
it looks like at the family level?
based on, like, the biological classifications of family? So they're saying, like, you know, what are the odds that all of the species in a current family trace back to a common ancestor, for just one common ancestor per family?
Right, but they're assuming, right? They're either assuming that all shared genes had to come from they're assuming that all shared genes had to come from a common ancestor. So they're assuming
not all shared.
they're assuming that beyond the family level or beyond the species level, you wouldn't share any common genes.
No. That is not anywhere in this paper assumed. That's not at all what this paper is saying. They're not comparing, like, the shared genes. They're comparing shared differences, like just genetic differences between base pairs. And so, like, there's absolutely no consideration to say that there's, oh, well, why should this family have this gene and this family have this gene? That's not what they're doing. They're saying, like, hey, there's a mutation in both of these genes, and it's in this base pair. And what are the odds that both of these families would have this mutation in the same place?
Okay, and the reason that in particular thing is a problem is because you're assuming common ancestry, and here's why. Because in common ancestry model, everything has to happen by mutation, right? So every change, every difference comes about by mutation. But if you don't assume that the differences are from mutation, variations could be natural variations, beginning variations. At the base level, we're talking about chemicals that are identical. So at the base level, we cannot identify what is a mutation and what is not, unless we're comparing things that are like Y chromosome and mitochondrial DNA, which can only really change by mutation. Otherwise, they're passed down directly. But you cannot assume, right? You're assuming that these variations came about by mutation, and that is the problem. So once again, you don't know what the separate ancestry model looks like.
Yeah, but we agreed at the start of the conversation that we both agreed that mutations happen. So you are agreeing that
Absolutely.
you're agreeing that
Yes.
many of these differences are from mutations.
Yes, some of them are. Some of them are.
So what are
But
the
I'm
other
not
ones
agreeing to the number. I'm not agreeing to the number, and I'm not agreeing to which ones are.
So what, like, with the example that Sal brought up earlier, where he'd seen different pseudo genes between humans and chimps that are accumulating mutations that are the same, in that example, are you saying that those aren't mutations?
Can I answer that one since he referenced me, Rebecca?
Sure.
It was only two nucleotides. So either, like I said, a creator could do that, or alternatively, it could be a natural hotspot. And we don't know a lot about how DNA may change. The evolutionists keep putting this random distribution, and I've already pointed out examples. You can't universalize that random distribution, especially like with histones. And you erroneously said, even after I cited the histone deletion mutants, that conservation is due to natural selection, or so-called mislabeled natural selection. So the other thing is, as I just looked at this up, statistical evidence for common ancestry, they totally ignored the point that I was saying. The nested hierarchies that co-evolve will not work if you're actually looking at the binding interactions and things like nuclear localization and transport processes. Evolutionary biology totally ignores that. And Dr. Dan knows that is he suspected this is a problem. It's going to come to bite you guys up in the next 10 years as we do more sequencing, as we do more protein testing, as we do more biophysics and biochemistry. That is something the evolutionists, all they do are these phylogenetic trees. They don't analyze the biochemistry, which is so important for these things to function.
I can tell that you really, really want to move on from the subject that we're currently talking about, so you can talk about your thing, Sal. I'm happy to do that.
That is the thing that is your undoing. You're trying to focus this.
Well, let's talk about it then.
Genetics.
Let me address.
This random mutation. I'm pointing out the variation cannot be random. This is totally on point. Why?
Okay, so then let me address that.
This is why it won't work. This is why this paper is invalid.
Okay, well, then let me address it.
If you just want to just cherry pick,
I'm trying to address it, Sal. You just won't let me.
That's fine, but I'm pointing it out to the audience. They're neglecting basic biochemistry that's so obvious that even a senior. I'm not neglecting it. I'm trying to address it, and you're literally talking over me.
I'm trying to address it here. Okay. First of all, you agree that, okay, he's not listening to me because he's still talking. I don't know. Okay. Sal, do you agree that humans, chimps, and guinea pigs all have broken gulo pseudogenes? But for some reason, you say that it could be a hotspot. Why chimp?
Say maybe. And you're totally ignoring all the big things, the elephants in the room. You're straining at gnats, letting camels through.
Okay, you don't want to talk about.
I'm not going to be playing with the little gnats here. You need to deal with the elephants.
Okay, hold on. Let me say something, Grayson. I'd be happy to talk to you about the gulo pseudogenes sometime after I am able. If you can present the evidence, and then I'll look into it and see if you're ready to present that evidence tonight. If not, we could do it another night.
My point was just that in response to, because Sal brought up a lot of points there. I was just trying to respond to the very first point. Okay, I'm just trying to address Sal's point here. Okay, he said that this could be due to some sort of hotspots where certain genes are prone to mutate and in a certain loci. And I would just point to the fact that humans, chimps, and guinea pigs all have broken gulo pseudogenes, but they're broken in the same way for humans and chimps, and they're broken in a different way for guinea pigs. So that would seem to be counter evidence to the hypothesis that these genes are just prone to mutate in certain fixed ways. Now, I would like to address Sal's other point about this whole co-of evolution thing, and that if you have just one mutation here, it breaks the whole system unless you have all of these mutations simultaneously occurring. Right? That was his point, that he's saying that's the big elephant in the room, right? Or am I getting that wrong? He's shaking his head like I'm not summarizing the problem.
It's not a single mutation, but if you have sufficient changes, it could be, because one mutation could be a silent mutation, right? So you need at least a few amino acids to change, but it has to have a corresponding change elsewhere. This is so basic biochemistry, it's unbelievable. They're ignoring it. And it is I disagree that what I said is derailing, because this is on point. This is the thing that falsifies all these papers and this nonsense that random variation can account for this. You cannot be changing binding interactions randomly and expect that you can randomly change one side of a binding interaction randomly and then the other side. That doesn't work. We know that in the pharmaceutical industry as we're trying to make, man-made zinc finger proteins. We are starting to know these problems. You guys totally ignore what's going to undo all of this. I can understand why you're going to ignore it. You're going to focus on the gnats.
I'm really not ignoring it,
because this falsifies it. And you're going to lose this in 10 years. In 10, 20 years, you'll be gone. Just like this whole thing about...
Sal, to be fair, you were saying that 20 years ago. But I'm not ignoring this. All I was asking for was for an affirmation that I summarized your point correctly. I did not say this 20
years ago. I did not say. You're making stuff up on the fly like you often do. I'm calling you out on it. You just made that up. Thank you.
Okay, can I just bring us back here? Sal, look.
Wait, I never got to address what Sal was saying, though. I really would like to.
Okay, go ahead.
I was trying to address it, and I was just trying to make sure that I was summarizing the problem correctly. And I was just asking for a yes, Grayson, you summarized that correctly, or no, Grayson, you got it wrong. That's all I was looking for from Sal there, and he didn't even give me that. But okay, look, for this specific example, Sal, if you had a gene duplication, so that you have one version of the protein that helps to bring proteins into the nucleus, and then you had another version that was a duplicant that then inherited mutations, it would still be functioning because you would still have a functioning version of that original protein. And then the second version is free to mutate and explore a range of other interactions. Duplications, right?
That won't work for changing the binding interactions. So no, I disagree with you.
Why not? I mean, if you have a duplication, you still have one version.
You explained the whole thing, so you can go back and watch.
You're going to ask me to try to explain that in 2 minutes. If you don't understand why it's problem, that's the whole problem. You think gene duplication can change binding interactions easily.
Well, you still have one working version that is like, so you still have the original system unaltered. So it's not.
Look, we can look at this. This is going to be an hour-long talk, and I've discussed this. You can't do that with a zinc finger protein when you mutate its binding partner. This is nonsense. That either that zinc finger is going to go to the wrong promoter region or some random region, or vice versa. It's not going to bind. If I have to walk you and spoon feed you all the little details, I'm happy to do that, but maybe not on this show. Thank you.
Okay, Sal, I don't need to get into an hour.
Sal, are you saying you're giving evolutionary biologists the zinc finger? Are you giving them the zinc finger again?
Yeah, I gave. I can't believe you saw that post. Yes, zinc fingers are God giving them the finger. Honestly.
Okay. Just FYI though, if you have a duplication event, then you have two versions of the gene. So then the original interaction is still occurring unaltered.
Make a new binding interaction. I'm sorry.
Yeah, but Sal,
it's go to random
the original,
but not be functional. Do you not understand that? If I randomly mutate a zinc finger, okay, so you duplicate a zinc finger, now you start randomly mutating its copy. Okay, at worst, it's just going to start jumping all around and maybe binding to random locations. How does that solve your problem? Do you not understand the basic biochemistry of that?
Yeah, you're only looking at the new version, right? You still have the original version unaltered, carrying about as normal. So that solves your entire problem.
No, it doesn't. You're making stuff up. I can make an hour show to call you out on your usual makeup. Anyone who actually understands the operation of binding interactions, specifically zinc fingers or any other thing, would know that that is nonsense. This is the same nonsense that I see from evolutionary biologists. They think in their mind they have solved this problem, but when you actually work the details out in detail, it starts to fall apart. I see this nonsense being even promoted in peer-reviewed literature, and guys who actually work with this, a lot of them will just laugh at that nonsense. So I'm happy to do a show on this. And look, I need to show diagrams. I need to show how a zinc finger works. I need to show all the ribbon diagrams and the zinc and how they interact with the promoter regions. I cannot do this in just kind of yapping and trying to refute your points. The problem with you, Grayson, is you leverage Brandolini's law. It takes 40 times more effort to refute BS that you generate. And I'm happy to address it, but maybe not in this show. And people know that I have talked about this a long time, and I've never seen an evolutionist refute it. And your gene duplication hypothesis, I'm happy to demonstrate you're wrong. Just like I demonstrated you were wrong about the amyloid hypothesis and your claim that there's no plasma in space. Thank you.
Look at this. I mean, he just accused me of Brandolini's law and then brings up all these separate topics. Like, I mean, come on. I mean, this is pure projection. Yeah. But basically, my point is that in a gene duplication, you still have the original system unaltered, right? You haven't thrown a whole monkey wrench in the whole system because you have a duplicate. The original system hasn't changed, so none of the binding interactions have changed either. And you just have another duplicate that's capable of exploring other interactions and accumulating other mutations. This is like basic gene duplication. This is how it works. But I'll just also point out all the different times that we have seen exactly what Sal is saying that we can't see here with these, like, basically this is like, you know, Behe's whole concept of, you know, oh, it's there's too many differences that would have to accumulate simultaneously. And then we've seen literally systems that match his description that you look at it and you'd think, oh, there need to be 4 mutations happening at the same time in order for this change to happen. But we've seen such systems evolve step by step.
Like what? Can you name one, Grayson?
Yeah, so the example that I like to bring up is this this self-replicating RNA system where they started with one RNA that was self-replicating, and by the end of it, they had 5 different RNAs that were irreducibly complex, right? If you took out one of them, the system would no longer work, which fits the definition of irreducible complexity, but we saw it arise from a system that only had one component, and it evolved and mutated into a system that had 5 different components, each of which was necessary for the system to continue replicating.
Okay, so
do you have a source for that?
Yeah,
I will find my source here.
Okay, so with gene duplication, consider you have a lock and a key. You make a duplicate key. Now I just take a random file and just file it away. Now it's free to mutate, so to speak. Now, do you think it's going to just randomly start being able to? So what you have to do in this co-evolving nested hierarchy, not only do you have to account for the fact you've randomly mutated this key. So you make duplicate key, both of them can turn the lock. And then you start filing down and just totally changing this one of the duplicate keys. Do you think it's going to just randomly match another lock with a precise, with a precise interaction? That'd be, at best, you hopefully don't harm the organism. And then so now you're going to say, okay, then some other lock evolves to match it. And really, it should be a series of locks and keys, where you have to open one lock to get to the next key, and then that key opens another lock. And you're going to play that game all the way around. And then you're going to say, somehow the original one that was working just disappears. Do you see how ridiculous that is? At the very least, you can just keep saying it in this discussion, but you guys never even modeled the difficulty from first principles. This is going to get you guys.
So, first of all, I thought you were talking about mutating the locks, not mutating the keys, but it doesn't really matter. But even so, there are multiple different locks, okay? And these things, first of all, are not as precise as you're trying to say here. At least most of the time, these are quite robust interactions where you'll have some variants that are slightly more efficacious or slightly less efficacious. It's not anywhere near like it has to be a lock and a key specifically. It has to be close enough to fitting in the active site, right? But there are multiple variations. Usually proteins and enzymes are very robust in their structure, like in terms of their interaction. So once you start messing with for your example, this duplicate key, first of all, you still have the original key, so you can still do the function and get the lock. This other key, like you said, starts changing in ways that are not affecting the original lock and key because you have duplicates. So I don't how does that not solve your problem?
It doesn't solve it because you have to change the lock and the key, the original lock and key. That's the problem of co-evolution. You have to use that mechanism to explain why the original lock and key changed. I'm just giving it to you. If you're not getting it, I understand because we're just doing this. But I, if you diagram this out, you would see the problem. You're thinking too much in gene duplication land, which doesn't work to solve the problem. This is so obvious when I was presenting this to faculty. This is so obvious, and you don't get it. I get it. I understand it. Maybe I didn't explain it that well. But this is, by the way, Rebecca, this is going to be a topic. I got invited to a Discovery Institute private event, but I'm free to give my public version of that lecture. I'm going to be covering this problem. It's a severe problem. It's gonna come and get you guys, and it's gonna falsify all of these papers you're citing and talking about random variation and evidence for common ancestry. The problem is if you assume common ancestry, these problems are going to be there and they're not going to go away.
Okay, well, we do agree on, Sal, look, I think their problems are massive and they go way beyond. But, we were trying to talk about this one specific thing because I do want to give a fair hearing. And so, but maybe we've exhausted the point. I mean,
Rebecca,
Grace,
can I bring up the chart that we were on last time that you said that we could maybe go back to, the insect evolution?
Sure, but but let me bring up first the paper that you cited. Okay, so, and my goal with doing this is that I'm giving the evidence for evolution a fair hearing. Okay, so this is what I'm saying. Grayson, I am taking this paper that you cited as the thing that you said about RNA, and I can't respond to it right now, but I'm going to read this paper and then I will make a decision of whether I think this is evidence for evolution, and I will provide
I really don't think that that's the right move, because I think that you're divorcing it completely from the context of which I brought it up. Like, if you're trying to say that I'm citing this paper as evidence for evolution, then that would not be accurate. That's not what I was doing.
What are you citing as evidence for? I mean,
what
are you trying to do with this paper?
This paper is demonstrating that irreducibly complex systems can evolve from systems that were not irreducibly complex in that same way. That's all I was trying to illustrate with that paper.
Okay. But I didn't mean to take it further than what you were saying. If that is able to happen, then I would give a point on the evolution side. Right.
Okay,
so that's all I'm saying.
I really think
you don't want me to award a point on the evolution side for that?
No, I think that the first paper that we talked about is enough to just demonstrate that evolution is a reality. So focus on that paper, or on the other thing that I actually is what I brought for my evidence for evolution, which is what I wanted to talk about, which was the insect evolution fossil record phylogeny congruence.
Okay, yeah, then we can do that, because the rule that I've made, because I've realized if people come on and they share too many different pieces of evidence, then I have too much homework to do. Yeah. So one piece of evidence per person.
Yeah, my only evidence that I want to bring is the same evidence that I wanted to talk about last night, because I only brought up the paper because you guys asked me to provide the paper. Okay, and that was not planned. The evidence I wanted to share is what I'm currently sharing my screen with. And I heard some of your responses to this last time, Sal. I'd be interested to hear what you think about this chart. But also, I was incorrect about something that you had said last time, where you saying that, and just so I can explain this to Sal here, we can just totally ignore this millions of years things. I mean, this is just like the order that these things are found in the layers of, we don't need to argue about millions of years here. We can just say, these layers are deeper, these layers are like more recent, whatever whatever words you guys want to use if you want to avoid millions of years. The fact is, these are like the layers down here, Devonian and Carboniferous and everything, and these are the more post-Cretaceous, post-Mesozoic layers up top. And you had said that you would expect, and by the way, the thickness of these lines here is the number of different species in each one of these insect families. Now, Rebecca, you were saying that you would expect for these lines to be getting thicker over time like this. But that is, I want to just, last time I just gave that to you, but I was thinking about it later. I'm like, no, wait a minute. No, there's no reason to expect that in the creationist model, because in most creationist model, they believe that these layers were laid down by the flood, right? So there would be no reason to expect that the layers down here would have less different varieties of species than the layers up here that have more varieties of species. But the main,
Okay. Yeah,
the
I
main,
see okay.
the main evidence that I think that this shows is that when we arrange these different families of insects according to their genetic relationships, right? This is a phylogeny. So the ones here are more similar to the families next to them, genetically similar, and they're least genetically similar to the ones way over here. We see the genetic relationships between these families reflected in the order in which they're found in the fossil record. Now, I'm trying to think of any possible explanation for this data other than evolution, and I just cannot think of any possible alternative. Like, water cannot sort things according to their genetic relationships. So this one chart I see as completely, just totally blowing creationism out of the water and eliminating it as an option of possibility.
Okay, well, remember, I do want to register, and I did mention this last time, but like you're using the word genetically, but you're talking about things that are, supposed to be millions of years old that we can't actually measure their genetics.
No, I'm not.
We can only look at their morphology. We can only look at their morphology.
I'm not talking about that. I'm talking about the genetic similarity of these families of insects. These families of insects are alive on Earth today, and we can genetically sample them and compare them.
Okay, so you're saying even if they went back, in your view, basically 300 million years ago, in your view, they're the same organism that was there 300 million years ago.
They're the same family, right? They're different species, but they're the same. Like if you find, like a grasshopper down here, right? It's in the same family as the grasshoppers up here and the grasshoppers today. That's just the law of monophyly.
Right. So the thing I want to say about the sorting is that the things that are at the bottom are also sea creatures. Now, I realize that that can be. Aren't those
these
sea
are not
creatures?
sea creatures? I don't know. And this is a dragonfly over here, and these are close
okay.
to the
I'm looking at the brown.
Okay.
So you're saying that's an insect?
Yeah, these are insects. Yeah.
Okay.
These are all insects, all of them. And the interesting thing is that these insects, so the way that they're separated here is by how they metamorphosize. So the ones over here have 3 distinct life stages where they go through like a cocoon, right? A chrysalis, and they have a larval stage. And the ones over here, they don't have this kind of grubby worm-like larval stage, but they still have some sort of a metamorphosis that they go through. And the ones over here don't really undergo metamorphosis. And the fossil record is sorting them in order based on whether or not they go through metamorphosis. And so isn't that weird that if it was a flood, wouldn't we expect some caterpillars or some things that can't get away very well to go at the bottom, but the dragonflies that can fly would be higher up? But that's the opposite of what we see.
Yeah, it's an interesting point. So let me say that it's an interesting point. And so, yeah, I mean, but what is also interesting is how many of these go down so low in the layers, right? Like, I mean, look at the difference between, you said the ones that can metamorphosize, like you go through metamorphosis. So I'm seeing those are reaching down to the layers that are 400 million years old, right? So how much are you saying that it's like all of them, like even the ones that don't go through metamorphosis? I don't know. I need to think about what you're saying, because maybe I'm not fully understanding your point. But I mean, what
The
I'm
point
seeing
is that
here is that
this
all of
data,
these families are going down very, very deep.
not all of them, but yeah, like some of the ones. But the point is that the ordering at which we find these insects in the fossil record is something that could not be accomplished by a flood. So like I don't know what the creationist answer is to this data, but I cannot think of any way that you could get a fossil record that looks like this, that matches the patterns of genetic similarity we see today, other than if evolution happened.
I do have a data point. We had found bacteria in amber that was dated at 200 million years old, and it looks like the bacteria today. And the evolutionists have dismissed it because it doesn't fit their model. So
What do you mean by looks like genetically, or what? What are you talking about?
Yeah,
I don't think that you could sequence anything that's that old.
They did.
Like,
okay, so you can look this up. I'm just throwing this out for Rebecca's sake. There's data that's just ignored that contradicts.
I would love to look at this data.
Sure.
Because
so this is showing. I'm sorry. Go ahead, Grayson.
Well, I was just gonna say, like, you'd understand, right, why creationists lots of times like to point to horseshoe crabs as being something that looks like it hasn't changed in millions of years. But I just want to make sure that the creationists understand exactly why we would expect for things that have achieved a fitness peak and have been optimized for their niche in the environment to not change over millions of years, unless that something in their environment has changed that forces them to change. So like a horseshoe crab is still functioning great in its niche, and its niche hasn't changed much in millions of years. So why should it change? And it's the same for bacteria.
I'm afraid not, because if you accept the neutral theory of evolution, that all falls apart, where most molecular changes are neutral. That is so you have the ultra Darwinists who'll give that story to explain it, and then you have the neutralists that are like, well, I don't care, but it is relevant. So this is the thing I see a lot of cherry picking data. They ignore data points that would overturn their theory, and creationists do that too. But I'm not saying, but I'm pointing out the evolutionary and paleontology community has done a lot of this too, and it's suppressing stuff. So we may, even if we didn't believe in a creator, there is a problem with the dating of the fossils and the layers. But anyway, I think Rebecca's sympathetic to this. We'll give you a little point for what you brought up.
I'm not sure. Okay, I need to think about it and really look at it, because what I'm seeing now, I'm looking at it further, Grayson, and what I'm noticing here. Oh, we have Immutable Destiny coming on too. Hey, Immutable, good to see you. Welcome on. I have a few things that I want to say about this, but like,
wait, really quick.
Yeah,
because I do want to hear your take on this, but there's just one thing that I wanted to say about Sal bringing up the neutral theory, and if we're going to move on to another topic, I would like to address it before we move on to something about the chart.
Okay.
Which is just that the horseshoe crabs of today would have very different genomes than the horseshoe crabs back then in the evolutionary theory. It's just that their overall anatomy would not have changed very differently. Like, again, they've accumulated a lot of neutral mutations, so their non-coding DNA would be very different. If you were to genetically test them, they would test as a totally different species, but anatomically, they would be very similar, and they would have very similar genes that were, if those genes were under very conserved natural selection. So no, that it doesn't. It's not just that. Oh, the neutral theory people are in opposition to evolutionary people. That is not an accurate depiction of the case. That the horseshoe crabs would be continuously accumulating more and more neutral genes, and their genomes would be very different, but their key genes and their anatomical features would not be.
Well, that was falsified by the bacteria found in amber. So
well, have you provided a source for that yet? I'm looking and I don't see anything in the private chat. I'd love to see that data.
Okay, while Sal is getting that, if he wants to, I want to bring us back. Can you guys see my cursor on the screen or no? Probably not. No. Okay. So if you look at the third line over, the first one in the blue.
Yeah,
okay?
This one.
And yep. Thank you. So that one, you're saying this, it's not one that undergoes metamorphosis, right? And it's lower down, right?
Well, it goes under like a hemi-metamorphosis. These are the ones that don't undergo metamorphosis. This one kind of goes a sort of metamorphosis, and this is true metamorphosis in the yellow.
Okay. But look at how far down now. Go to the first one on the yellow.
Mm-hmm.
And it goes down to the same point. It actually goes
not exactly
lower.
the same, but pretty close. Yes.
Lower. It actually goes lower.
It does not. It goes down to here, which is just above this dotted line. Let me zoom in here. It goes down to here, just above that dotted line. This is the dotted line over here. So it's not close, but I'll grant you that it's close, but it's not. It's not going down lower.
Okay. Well, so anyway, when I look at this right now, I mean, I'm seeing you're making an interesting point with the one in the brown. Okay, it's an interesting point with the brown because that does seem to go down lower than any of the other ones. And you're saying those don't go through metamorphosis, right?
Mm-hmm, yes.
Okay, so then I will say that's a point for, okay, it's showing a difference between the organisms there. But one of the issues that comes up is when I'm looking at this chart, I'm seeing a lot of separate ancestry, right? And there's lines drawn to link these things together, but those lines are not represented by organisms. So we're actually assuming that there were transitions, but what it actually looks like is a lot of separate ancestry. So, when I'm looking at the fossil record, look, I can see some things that are problematic for young Earth in the fossil record. Okay, fully admitting lots of problematic stuff for young Earth in the fossil record. But it's also, the fossil record is extremely problematic for evolution. And this is one of the reasons why. Because as far down as we're looking, we would expect to see, if these things are undergoing millions and millions of years of slight modifications, we would expect to see not sudden appearance of insects that have full wings. We would expect to see a very big progression. But here, what you're showing me in this chart is fully formed organisms that have been around for 300, 400 million years, and they're still around today, and we don't see them progressing.
No. So there are varieties of the different species within each of these families. They're not all identical, like you're implying. But I also want to make it the point that evolution does not predict that we should ever see half-formed organisms. Every organism is fully formed. That's not a prediction. That's not something that we would think to see in evolution. Are you conflating like creatures with transitional intermediate anatomical features with not being fully formed animals? Like, I don't know what you're trying to say when you're saying that, oh, well, we're only seeing fully formed organisms, so that's somehow contradictory to our evolutionary expectations. I don't see that at all.
Well, I think it's a little strange that everywhere we look, all the way to the bottom, we find organisms with complex systems that are fully formed and functioning in an integrated way. And we don't see them transitioning to other forms, except in very rare cases where you can say something like a Tiktaalik looks like it might be a combination of, things, but that's very rare. But we've got to get from the first living organism to the diversity of life today. but everywhere we look, it's like, boom, wings,
Yeah.
boom, eyes, boom, all these, fully formed systems. Well, how did you get to the eye? Why can't we look and see, oh, poor little guy, he almost had an eye, but he didn't quite make it. Why do we see fully formed eyes? Why do we see oh, poor little guy, he tried to evolve himself a wing, but look at it. It just wasn't flight worthy. It was just
Because
like a little nub.
because that's not how evolution works. You don't just like go from a nub to a wing or like a half eye to an eye. Like that's like we do see.
How does it work, Grayson? How do you get to an eye?
Like each intermediate step has a function. That function doesn't necessarily need to be the same as a wing's function or an eye's function, but you take functional parts and adapt them to other functions with small mutations. And each intermediate is fully functional. You're not going through any half functional intermediates on a scale in the fossil record. So you can, on like a biochemical scale, you can get like neutral mutations that just aren't deleterious enough to get purified by natural selection. But on a scale like this, I mean, in the fossil record, I would also say that it's not very rare that we see transitional anatomy. We see it all over the place. You could pick any two clades today. Like, I mean, this is another point that I think is one of the stronger evidences for evolution, is that if you pick any two clades that are neighboring in the fossil record, you usually are able to find fossils that have intermediate anatomy to both. So the famous examples are dogs and bears, neighboring clades. You go and look in the fossil records. When you stop finding dogs and you stop finding bears in those layers, what do you find? You find dogs that are more similar to bears and bears that are more similar to dogs. And then you go back further and you find like fossils that have even more generalized anatomy. So you can pick any two neighboring clades in a phylogeny, and you can go and find fossils that have intermediate qualities of both. Now, that doesn't make any sense under a creationist model, but it's exactly what you would expect for an evolutionary model.
Well, I challenge that, but I can't challenge the entire thing today, but I can challenge the model that you're putting up here. So let's look at this and see if we can find a transitional form to metamorphosis. So how do you expect, if this is showing progress to metamorphosis, where does it go from? How is it changing? Where's the change?
So you mean like, what is a transitional form between like holometabola and?
Yeah, let's do the Hemiptera and the Hemiptera. The purple and the yellow. Let's go the purple and the yellow.
Okay, so holometabola and pterygoda. Okay, well, I can certainly try to find that. I don't have it off the top of my head. I don't know everything, but I'll try to find it.
Well, let me tell you the problem with trying to find it. And you're welcome to find it another time and then bring it back on as evidence. But I'm going to tell you a few problems you're going to have to deal with. Number one, a caterpillar can't reproduce. So if an organism is going to become a caterpillar, these organisms over here are going to become a caterpillar. They have to, in the caterpillar, at least with butterflies, and I haven't studied all of them, but basically it entirely breaks itself down into goo when it goes into the chrysalis. So you're going to have to get a complete body system change. When it comes out, it has a totally different body system. So you're going to have to figure out, how did it get to this transitional form where it was able to build itself into a caterpillar with a plan to go into a chrysalis, turn itself to goo, and then come out with a different body plan. So how does that happen? Where in the world do we get that? Because the caterpillar can't reproduce. So if it starts out as a caterpillar, it doesn't have millions of years. It's got to get into that chrysalis right away, because it can't reproduce unless it gets in the chrysalis, turns to goo, and comes out with an entirely new body plan.
Well, by the way, the figure that I was sharing is from a paper titled "The Evolution of Insect Metamorphosis." So the paper
Cool.
is
Let's
addressing
read it.
exactly that.
Yeah, let's read. Let's make that a homework. I'll make that a homework. Actually, I was pretty interested in that when I saw you pull that up last time. So I'll make that a homework project, although my homework is kind of stacking up here, but I will make that a homework project. Okay.
I'd like to add one thing.
Sure. And then let a Mutable talk after that. Yeah.
A lot of evolutionists love the fossil record because it erases all the important details. You have to deal with stuff at the molecular level to argue these changes. And when you have large protein, you have major new protein families that are needed to make these changes, which we haven't even sequenced, we don't know, we haven't even characterized it. But once you have to start to account for that, the problem starts to become really difficult. That's why I'm really glad Dr. Dan conceded. All major protein families do not have a common ancestor. So to the extent that you might have to have a caterpillar that can now become a butterfly, we don't even have an accounting of all the new proteins that have to be there, that just have to sort of pop into existence. Just looking at the fossil record and saying that evolved is not a solution to the probabilistic issues that arise in building proteins with binding interactions and all sorts of functionality. The more that we know, the more problematic this is going to be. So this is not totally fair to be saying, hey, this just happened because they look kind of structurally similar. But when we know that when we start to evaluate things at the molecular level, the story starts to fall apart, and that's going to continue to happen. I definitely saw this with Change Tan. She was an atheist, communist China. She studied molecular biology. She saw the transition between prokaryote to eukaryote. When she did the accounting, she said, no, evolution's not a good explanation. I'm predicting a similar thing will happen as we start to gather molecular data. We're going to see all these orphan genes and these taxonomically restricted proteins and systems that just don't make sense from a gradualistic standpoint. And I'm sorry, Immutable. I should have. Hi. I'm sorry.
He just left because he had to reset his microphone, he said. But basically, I want to say that when you were saying that you're making these predictions, that is the thing that is so devastating for creationism with the fossil record, is that we're able to use evolution to predict the kinds of anatomical features that these fossils have before they're discovered. I mean, I've never seen a creationist successfully predict the anatomy of a new fossil, but it has happened over and over and over again with evolution. I mean, going all the way back to Darwin predicting Archaeopteryx 2 years before it was found, to Tiktaalik predicting the exact anatomical features in the exact layer that we found it. I mean, how can you possibly explain the successful predictions of evolution in regard to the fossil record? And I grant you, I'll just grant you, Sal.
I'm going to give you those points. I'm writing those down as points. Tiktaalik and Archaeopteryx. I will write those down as points for the evolution side.
Well, there's a lot more than just that. I mean, they predicted synapsid.
You can bring them. That's fine. But I want to give Immutable a chance to talk. I'm going to write those down so I don't forget to put them on my points for evolution. But Immutable, do you want to say something?
I joined because I was driving back from the gym and I was hearing Sal go on about this. I don't know, Sal. So let me ask you, what is your, I don't know, what exactly is your objection with gene duplication as a mechanism for evolution? Or do you view it as a problem? Or I heard bits and pieces of it, and I think several years ago. But
first of all, greetings. So the first problem with gene duplication is even more basic than what Grayson pointed out. If as Dr. Dan has agreed with me that proteins don't share a universal common ancestry, then gene duplication will not explain that. So that's out of the way.
Okay, before we go on,
do we agree on that? Gene duplication will not explain the emergence of these.
Well, that's the thing. Okay, so I want to clarify that, because to my knowledge, right, there's no expectation that protein families should share a common ancestry. Well,
we'll see. Then we're all on agreement. But
yeah, so
why are there no papers pointing this out? How is it so blatantly obvious? How come creationists don't know about this? Maybe they should. So I'm glad that you're agreeing that it's not, that the evolutionists are admitting these major protein families just kind of, we don't have an explanation where they came from. We don't know what their original ancestor was. We don't know the process of how they emerged. And yet they're so amazing, like the cryptochrome protein that enables, quantum entanglement to do navigation. I mean, that's just amazing. I mean,
sure.
You could just say, okay, it just kind of popped up, or it gradually evolved, and you never work out the details. But so we're in agreement that gene duplication won't explain that. Is that correct?
Yeah, I mean, I
Okay,
think part of that, right, but I mean, I think part of that is erroneous, though, because what you're saying is that evolution needs to explain why exactly why protein families should be genetically related.
No, that's a mischaracterization. They need to explain how those families emerge, especially the really complex ones.
Right, but in that case,
right. So the ones that the proteins. I'm sorry. Go on.
No. So in that case, I think it becomes a question more pertaining to the origin of life. I mean, I think the big issue here is a question of the question you're fundamentally asking is about the origins of protein families, which is a deeply related question to the origin of life.
For some protein families, not all. Collagen was a late addition. So
right,
were zinc sugars. We could probably name more. I just haven't surveyed all of them, and we're still learning. But I'm pointing out gene duplication will not explain that for the late arrivals long after abiogenesis. Another example would be all the proteins involved in spliceosomal, in the spliceosome, all the spliceosomal proteins, which is long after the abiogenesis event, and practically very critical to the evolution of eukaryotes. I could also same for the proteins of the nuclear pore complex. So if you're just going to say we all evolved from a single common ancestor, now you have all these problems that are not explained by gene duplication, and you can't move that back to abiogenesis. So I've just given you a set.
Okay, but I mean, if you grant, so I mean, you're at least granting the existence of some basic genetic of some basic Luca type organism. Do we agree on that?
Only for the sake of argument, and I'm
okay.
For the sake of argument, I would encourage creationists to just assume it for the sake of argument, even if they don't believe it. Okay, so I'm just coming clean. I believe in a special creation event, but I'm happy to work through the model that you have suggested for universal common ancestor for all cellular life, a Luca.
Right, but so in that case, I'm wondering in that case what kinds of and I don't want to be technical here, but how fancy molecular machinery do you think Luca had? What kinds of things could Luca do, as an organism that is at the most basic level? Because it must have been able to replicate itself. It must have been able to maintain its metabolism. How about
a bacteria or an archaea? Sure. I mean, look, I'll grant that. How about we grant that? I have no problem granting that for the sake of argument. So
right.
do you have a problem with me invoking a bacteria as kind of a Luca?
No, I think that's fine. I mean, I think bacteria, archaea, it's fine either way. I mean, my point is to say, if you're in terms of what's basic bacteria are able to do in terms of their organization, in terms of processes they're able to undergo in terms of genetic regulation and stuff like that, it's not inconceivable that if you have bacteria that are undergoing events, for instance, where it's better for a bacteria to develop certain or to allocate some of its resources for, say, part of its replication, right, and that involves the creation of some new organelle. You have parts in the cell already that are responsible for various things like cytoskeletal arrangement, things like this, right? And now it just becomes a task of reorganizing that and having that focused to a certain part of the cell.
Right? Well, the problem is when you get to the details, as Professor Tan, I mean, she was on your side, okay?
Yeah,
I tracked her evolution as a scientist, and she was on Rebecca's channel here. She said, look, I was teaching molecular biology. I just couldn't in good conscience say this happened naturally.
Right,
And
sure.
really
I'm not saying, I'm not trying to say we figured all this out, but I'm saying
really quick. I just got to go. Thanks for having me on, Rebecca. See ya.
Thank you, Grayson. Take care.
Yeah, good to see you, Grayson. But I mean, certainly, like these aren't things that can't that fall in the category of unfalsifiability either. I mean, you can take very basic bacteria, you can kill some of the genes that they have that make them sufficiently complicated, and you can try to test and see, okay, what kinds of things would need to happen for, say, a bacteria to develop some kind of organelle or something like this, right? I mean,
well,
these are things that can be tested.
Yes. The problem is if you have a nucleus where a lot of the transcription and replication happen, as in a eukaryote, you got to be able to shuttle things back and forth through that membrane, and that's a controlled process. And that's where you need nuclear localization signals. Those signals are like it's like a password.
But again, though, it's another kind of regulatory signal that's triggered by some kind of molecular event, be it phosphorylation, be it whatever. And I'm not a person who like, as my background, like I've taken biochem, I have a certificate in biotech, like, but I'm not a person like who's actively researching some of these areas. So I'm not like aware of like the precise mechanisms for some of it. Sorry. But the point is to say, if it's an issue of having things transport across some membrane, that kind of event already occurs within even bacteria.
It's in a controlled way. So let's say I mean very controlled. You would have to format all the proteins that are homologous, not to mention you have to make a whole bunch for the nuclear pore complex and the spliceosome parts. Hundreds of proteins, let's say even 200. I'm just picking out a figure. 200.
I don't know why it would. Okay, so I don't know why it would require. Like, I'm confused. Like, where the justification for that number is coming from.
Okay, so we know that there are about 8. The minimal cell, like say a yeast, the eukaryote, has about 85 in its spliceosome. The human spliceosome has about 200. So I'm picking out 85 there. The nuclear pore complex may have 100 or so. Angelo actually counted them. I didn't count them. Okay, it's not a small number.
Right.
So and then you have all the things involved in transcription and replication. So your topoisomerases, your helicases, your polymerases, the helicase loaders and primases, et cetera, et cetera, et cetera. That all has to be shoved into the nuclear pore complex. All of them need a password. They all have to have. These are so critical that at least a lot of them, if they're missing, the creature's dead. And so you have to simultaneously put this password that the important recognizes in all of these proteins that are homologous, one that are novel, and then one that are homologous to their counterparts in bacteria. The odds of that happening just seem kind of like too difficult to swallow for some people. And ChangeTan's an example, and you can kind of count me in that area, but I didn't need as much persuasion as she did. She's teaching that course. So that's all I can say. Okay, I'm sorry. Go ahead.
No, you're good. I mean, on one hand, if they're homologous, that's already. I mean, from an evolutionary point of view, it's a very likely indication that at least certain parts of this kind of molecular machinery was passed down, right? Maybe not on the exact line of bacteria you're talking about, but certain mechanisms that exist were passed down and were modified in some elegant way.
So this is where, again, the nested hierarchy and similarity. I am arguing, and I would presume ChangeTan is, that, they are homologous, but the change is enough and it has to be sudden enough that design is indicated, because those nuclear localization signal changes have to be there to begin with. Otherwise, it doesn't get through the nuclear pore complex.
Okay, but so my understanding, and correct me if I'm wrong, my understanding is when you have like transport of, say, a protein across some kind of I'm just going to call it organelle. That's not the right word, but
membrane. How about a membrane?
Yeah, membrane is
membrane-bound organelle. Okay.
But in a nutshell, basically, like what you have is you have some region of the protein. Usually, it's not essential for like the actual function of the protein. Usually, it's some part that's free to be grabbed onto by a protein that's able to allosterically bind to it. And then the other process that usually occurs once that's done is, if I'm not mistaken, you have a lot of, basically a lot of like molecular activity. Generally, I would guess it's like the consumption of ATP in order to effectively move it through the membrane. I mean, is that more or less like accurate to your understanding of?
I don't know that it's ATP either. There might be other, like the one with
Yeah, sure. Like
guanine. It might be guanine. Okay, but that's immaterial. The point is,
like, what do you agree
okay,
that?
So I'll tell you how we find this out. So when we're trying to look for the passwords, we actually do little knockouts to see if it's essential. So when we knock it out and it doesn't localize to the nucleus, and this is only like, anywhere from 5 to like 20 amino acids. We knock that out, the creature dies. It's like, okay, now we know this is where this little password has to be written into the protein. Right. Even if it's homologous, you have to figure out how you're going to get 14, let's say 10 amino acids in the right sequence and not just that one protein. You have to do like 400 simultaneously. Okay, let's say even 50 are critical, life critical. How are you going to insert a password that's 10 characters long and 50 proteins simultaneously? Otherwise, the creature's dead. Now, you can postulate some gradualistic mechanism, but that's imagination. That's not.
No. I mean, I don't know if you're talking about like. I don't know if now we're talking about tertiary structure of a protein, or if we're talking like. To be
it's
careful.
Primary structure. It's primary structure. Just the sequence. 10. It's roughly 5 to 20 amino acids. I've seen some as long as 25.
Sure.
It's just like when you're typing your password, even a 10-word password is kind of challenging. You could mistype it, and you don't get logged in.
I mean, generally though, the chances that like a given substitution for a protein ends up like completely throwing off its function are very, very small. I mean, because the majority, and let me justify that before you jump on it. The reason I say that is like the majority of like mutations that are going to occur are going to be changing like an alanine to a valine or something like this, right? They're going to be mutations that either that are missense or usually that are silent, right? And,
right. So let me try to deal with that. It's just like a car. There are parts that are really critical. You could modify a lot of parts in the car, no effect, but you cannot generalize that to the parts that are critical just because you could mutate. You could randomly change, like say your windshield wiper to that. And so there are things with specific binding interactions and also these are critical. And I do happen to know, because I work with Joe DeWeese, he identified a few spots. He said, look, if you change one residue here, it's dead. That would be in the metal binder region of the topoisomerase. So you can't generalize that. So I will grant that you can make variations in parts, and it's just going to be,
Well, sure, but in that case, I mean, what we're describing here essentially is that look, there's some aspects of the protein that can't be changed, and those coding regions of the genome that are responsible for that are under purifying selection by definition, right? Because if that kind of thing undergoes a mutation in which it destroys the function and the organism needs that function to survive, the organism dies. It's not going to pass down its genetic lineage, and game over.
Well, I don't like the word selection. I'll grant it. It's misconceived, and it also doesn't explain the emergence of novelty. Just like I said with the nuclear localization signal, if you're gonna have importance that guard what goes in and out of the nucleus, if you don't have those simultaneous changes in the nuclear localization signals of all those other proteins that go in the nucleus, it's dead. There's no purifying selection.
No, I'm not.
I want to ask a question about this, though, since you guys know a lot more about biology than I do. So let's just suppose, I want to suppose that a protein morphs, okay? And it's able to change mutations enough to get a new function. Now, how would that, if I'm supposing if now, wouldn't not only that need to be changed in the protein itself, But if that's going to go on to the next cell, then wouldn't it also have to get coded into the DNA? So it would have to get coded into the DNA in its new form. Otherwise it's gone, right?
So to answer your question, if it's the case where a protein changes from one function to another, which I imagine probably is statistically rare, then yes, because it's a genetic change that's occurring. Although some proteins are, if I'm calling the term correctly, polymorphic, meaning so there are proteins that can do more than one function at the same, or that can have more than one function while it's around. So it might create some metabolic product, but it also might interact with other proteins in such a way where it's causing regulation for, it's regulating some protein in some way to slow down some process or speed up some process. But in the case where the change is in the genotype, yes.
Okay, so do you, in your vision of the origination of a new protein, are you seeing that happening? Do you envision that happening in the genetic code? Because it seemed like you guys were more talking about the protein itself once it's built changing. So
it has to originate in the genetic code. That's correct.
Yeah.
Okay, so it has
to change in the DNA. The genetic code is a different word. I'm sorry to be persnickety. Yeah, you're correct. It has to change in the DNA, and that's reflected it'll be reflected by the protein that the DNA codes for.
Yeah.
Okay, so then immutable, are you saying that because weren't you guys talking about the possibility of proteins morphing outside of the DNA?
So
Okay.
So what Sal was talking about in this case, if I'm recalling right, is something we're talking about, membrane proteins or like transport proteins or something evolving or something like this.
Well, it's the whole system that would implement the membrane-bound organelle, and it's not trivial.
I'm not saying it's trivial. I'm saying that. Okay. If you have systems which are already able to transport proteins, the kind of major thing that seems intuitive to me, at least, right?
Why would you assume such a system exists? I don't understand.
Because if you were around earlier in the kind of conversation,
no, you have to assume that.
Hold on. Because in the conversation we were talking about earlier, we had already talked about the existence of LUCA and basic functions that LUCA.
Yeah, but you're going way up ahead. We're talking about how about the how does that actually form in the first place? How does the actual functional system come into existence?
We're not talking about that. I'm not talking.
You have to. It's part of your paradigm.
No, I'm not, and I don't.
Okay, so
I did want to respond to that.
Yeah, go ahead, Sal. Sorry.
I was saying, look, this is like saying a VW Volkswagen has already all these processes. You can easily transmute it to be a Prius hybrid. It's like, yeah, it looks kind of okay on paper, and if you have engineers working on the problem, they can make that transition. But whenever you have to change the sizes of things and the way things connect, it's not as straightforward as you're suggesting. And this is what I do see in evolutionary literature. They don't actually work at the mechanistic details of what will actually go wrong. All the things that could go wrong as you're transitioning from one thing to another. So if I'm just changing the cylinder size of an engine to make a bigger engine, you can't just, it's like it looks good on paper. It's like, oh, that's, it already works. So why don't we just evolve it? It's more detailed than that. And when you get to the molecular level, that's what I'm trying to show. It's like, oh, that's just a few amino acids. I'm like, look, it's not a few amino acids. It's like 5 spread across like 200 other proteins simultaneously. It's not as easy. When you get to the accounting, it starts to become a little troubling. And I have read literature on eukaryotic evolution, and a lot of them saying we're probably never going to solve this problem. And I'm like, well, yeah, I see that too.
I don't know if I've heard them say that. I mean, I think a lot of it is that there's a lot of ambiguity in terms of what exactly the transition from prokaryotes to eukaryotes is. I mean, we have certain indications of at least how endosymbiosis occurred, which was a major step. But certain other parts, I mean, in some sense, it's irrelevant. Like, it doesn't matter necessarily how exactly it occurred. I mean, we know that it occurred. I mean, it's entirely possible that there's multiple different pathways to it, and just like one conversion one happened to be the one that life developed with.
But immutable, we know it occurred, but we don't know how it occurred. We're trying to figure out, can it occur by natural processes, or did a creator make it? So we're assuming a creator made it because that's our observation of the world.
Yeah, I mean, the reason my predominant issue is I don't think that creationism can make specific predictions which are falsifiable about how exactly the particulars of that process occur. Because if there's something which seems to not fit, it's a matter of saying, oh, well, God chose not to do it this way, or God chose not to do it in such and so way. And the reason, and again, so like, I'm conceding that I'm not saying it's a simple process. I'm saying that much like how when you're trying to solve a crime scene, you don't always have a letter of who done it. You have a lot of pieces of different fragmentary evidence that are scattered around. In some ways,
yet you would never conclude that the crime scene would be a natural process, right? You'd always come to the conclusion that somebody did it, right?
That trying to figure out of the who done it make more sense than others. And ultimately, like, what we're trying to do is we're trying to figure out which of these events are the most plausible. By definition, though, there's going to be almost infinitely more pathways that fail than those that succeed. And the ones that we have are the ones that succeeded, not the ones that failed, because they're not around.
Hi, Matt. Great to have you. And Sal wants to share a video, but I want to give Matt a chance to speak before we pull it up, if that's okay.
Sure.
Well, I just wanted to respond to some of the things he was mentioning before. I was not able to get on at the time, but so you were bringing up fidelity about, like, how things will transfer over, and how genes they're not as corruptible as we make the claim to be. The thing is, we have a lot of you made the insinuation that was definitely something I heard earlier.
I don't want to hear what I insinuated. I want to hear what I said, and I want to hear your response to
it. We can go back. You mind pulling it back up, Rebecca? It's maybe 10 minutes ago.
No,
you can just play the whole clip. It's fine. It was copied.
That's a little complicated.
If you have an exact instance of what I said and want to respond to that, I'm more than welcome.
Well, I wanted to point out that you're not taking into account things like the enzymes that repair, that actually go in and they functionally repair the genes. That's actually something that's never accounted for some reason in you guys' theory. And you need to really consider the fact that not only do those exist, but why do they exist? You shouldn't even expect something like that to just form naturally. Why would something that has, think about this, it's actually requiring forethought and understanding of your entire genome, just so we can go back in there and then fix the one that's wrong. Do you understand the issue with that?
That's already involving something that would have occurred prior to the development.
But you're already insinuating that there's supposed to be certain systems in place, but why would such a system even arise? Do you see what I'm saying? You're kind of giving yourself way too much stuff ahead of time, and then you even try to insinuate that Luca would have such a thing.
Look,
why would you think that such a system would just arise naturally? That doesn't make any sense.
Look, it's irrelevant to me what things you think I believe are a stretch too far and a stretch not. The basic explanation for why such a thing would occur is that if you have systems which don't have that capability and there's constant errors which are damaging the genome,
still have that capability, why would it arise in the first place?
I think I'd let you talk. Can I have the same privilege?
No, I'm questioning your assumptions. I'm trying to help you to see that you are assuming so many things before you actually make your statements.
I'm really trying to respect you, but can I please talk?
So I don't have the permission to call out your assumptions. I can't do that?
You're calling out my assumptions, but when I'm trying to explain it, like I was just now, you're interrupting me.
Right, okay.
I'm halfway through an explanation, and you just chose to dive in. And
so instead of having you go on a 10-minute rant about something that's irrelevant because it's talking about assumptions that you already made clear, that we
Matt,
said,
can
hey, these
I-
are actually kind of wrong. You shouldn't be assuming this to
make
begin
sure-
with.
Matt, can I make sure I understand something here? When you ask somebody a question, is the person supposed to give an answer before you ask a new question, or what? I'm just confused on like the basic art
If
of
the
conversation.
answer does not come with proper assumption, don't you think
then
that I should be calling that out?
My full answer.
Should I not be calling that out?
You need to listen to my full answer before
If
you
it's a
call
10-minute
it out.
rant, I'm not going to do that. I guarantee you that.
Well, it's not. I was barely talking for a minute.
Sir,
Let's
I've
let
listened
him
to you
finish.
talk for the
Let's
last
let him
good
finish.
while. What are you talking about?
You're asking me a question. You want an answer. I try to answer you. You interrupt me. I don't see what part of this is hard for you to understand.
Sir, I'm not in the business of trying to do a whole rabbit trail where you go off into what you think about something, but it's based upon wrong assumptions. I'm
You're
trying to
asking
help you
me
to
for
see
an
you are
explanation.
starting from a wrong premise,
Matt,
and you're
you are
supposed to
asking
backtrack
me,
and say, okay, well,
Matt,
maybe you're right. Maybe that is not the way I'm supposed to view this.
Matt, you are asking me for an explanation. I cannot justify the assumptions of my explanation before I've had a chance to complete the explanation itself.
Yeah, I think you should give him at least 2 minutes.
You're good.
Yeah.
That's fine.
Yeah, the basic explanation for why the existence of error correction occurs is that if those systems don't occur, and you have cells which are already capable of doing things they're able to do, but not error replication, as those mutations occur, it's going to damage parts of the genome that are responsible for basic functions, and eventually the organism will go extinct. Just on a probabilistic scale, that's an inevitability.
The issue, sir, is that those are required for the existence of life in general. Like
Right,
they
and I just
already have to be there.
Right,
But
and
you're
I just
assuming that
explained
these
to you-
systems just generated
I
without.
explained to you why such a system would arise rather than a system rather than modern systems not having error replication mechanisms themselves.
All you did was make a just so story and not
No,
actually defend it.
Matt, do you not agree with the explanation I gave?
Absolutely not.
Do you think that if organisms are constantly accumulating mutations with no way to correct those that are deleterious, they're going to survive?
If a code was accumulating
That's not the question
mutations,
I did not mention the word code in my statement.
I know that. I know you don't want it to be linked to that, but you know what it is.
I didn't use the word code.
Doesn't matter. It does not matter, sir.
Codes.
We can understand that it's functionally the same thing, and even
No,
Richard Dawkins
it's
admits
not.
as much.
I don't care what Richard Dawkins has to say. I'm interested in you answering the question I asked, not whatever you're interpreting that I'm asking.
I'm trying to check your assumptions. You're making a lot of assumptions and just so stories.
No, I'm not.
Yes, you are.
Very simple
Guys,
question.
I think this is a good point. We're kind of at an
impasse.
impasse. So let's let Sal show
Rebecca,
his video. Yes.
Rebecca, just for clarification, I don't mind having conversations with Matt, but when he's going to make insinuations and add things to what he thinks I'm saying, and then say, "Oh, justify this," even though it's not a thing I said, I'm taking that as bad faith acting and being disingenuous.
I guarantee we can go back and find the part where you talked about that if you want to continue acting sway.
Matt,
that's what's a little bit hard, as I don't remember the exact words that were used. So it's hard to.
A few seconds ago, he was saying I was making insinuations about codes. That word did not.
Well, you didn't use the word
No,
code.
no, I didn't say you used a code, but we both know that it is a code.
No, I don't agree.
Okay, well, you don't have to agree for it to be the case.
Yeah,
so,
Immutable,
are you saying then there wasn't a code? Like you?
I don't think that in the sense that we talk about normal codes, that the genetic code, that DNA is a code in that sense.
They've literally written books in DNA. I don't know what's making you think that it's not a code.
I'm glad you read Science American articles that say that.
No, I'm not. I actually read science research where they. Have you ever actually looked at the Craig Venter work?
What about it?
On Cynthia?
What about
Remember,
it?
he made the what he thought was like a synthetic cell.
Okay.
His entire team wrote a story and they put it in the genome. Did you know that?
That's news to me.
Well, I guess it must be. And here I thought that you were a biochem guy, right?
Yeah, I'm unfamiliar. You're mentioning one particular biochemist out of potentially thousands.
Well, it was pretty big. No, it was pretty big. It was all over the news for a while.
That Sal wants to show.
I'm sorry, Sal. I am interrupting. Go ahead.
Thank you, Rebecca, for letting me show this a little bit of a background. I
And it's
mentioned
only
that
4 minutes, guys. So, I mean, but
Sure.
can we watch it on double speed, Sal? Because I mean, 4 minutes is quite a long time.
I'll try to run it at double speed. I do want to say Change Tan was the atheist from communist China. It was the study of this that, in similar systems, that made her a creationist. So
complexes are the only channels.
It's really patchy. Are you guys hearing it?
Yeah, Sal, if you want to give maybe a
ions
succinct
and
explanation of what the issues
The
are, that might help.
macromolecules can travel between the nucleus and the cytoplasm.
There we go.
Yeah, there's a little delay on my end too, so the audio is a little jank.
Yeah, it's frozen now. Sal, it's frozen.
I stopped it.
Okay, what's the?
No, I stopped it.
Maybe you can go to the relevant slides and you can give an explanation if you want.
Well, I feel like we've already had this explanation. Sal's trying to show a video that illustrates what, his issue that he's been talking about throughout the stream. So, actually, guys, I would like to maybe do closing points because we've been going for quite a while. And so do you guys want to make closing points about what we've talked about so far, or just, you can say
sure. I mean, if you want to talk about if you want to talk about a different point, I mean, I'll go last if anybody else wants to make another point so that I can address it if need be.
Okay, if I could share my still screen, and I'll just finish out with, this is a still. This is the topoisomerase enzyme. This is the amino acid spelling. These are the regions. This is one way to look at some of the critical regions. If you knock out these DQs, protein's gone. So there are sensitive spots. In the red is a localization signal. That password has to be there, otherwise the creature dies. And my point is, you need these passwords in about at least 200 proteins simultaneously, because if each of those proteins is critical to life, it's dead. There's no reason to have, this is where you have the problem of a half-formed organism. In fact, it could be 95% there, but or even 99%. But if you lose that critical 1%, the whole system's gone. This is a probabilistic problem. And we'll see this issue emerge with all sorts of binding interactions. If you model it like logins and passwords, it's not exactly like a password. You have to get every letter right, but you have to get a lot. It's going to cause a problem for the nested hierarchy. The evolutionists do not even have a clue how severe this problem is, and I'm going to raise attention to it. And this is probably the better way to attack the nested hierarchy. I just had to weigh in. I said there are ways to attack it. I'll just say the nested hierarchy exists. There's some subtleties to it, and it has to be via common design, not a random process. It'll fail with a random process. Rebecca was right to point out there might be a few things we would concede that we don't have explanations for. But I wanted to point out, if the evolutionists keep saying, well, we're working on the problem, then creationists should be entitled to the same. We don't have to prove, because if we are insisting that the evolutionists are held by the same standard, their whole theory is unproven almost at every major level. So I find it ironic that they're going to be going after us for little gnats when they're letting camels through. Thank you.
Thank you, Sal. Okay, Matt, I want to let you go next because I want to give Immutable the last word on this, and I'm not going to argue with Immutable, so he'll get the last word, but I'll just say some final thoughts. So go ahead, Matt.
That's fine. No, I really just came in to try to help him see the issue with the reparative enzymes and how those were necessary for life, and that's actually part and parcel with what Sal was talking about, because he gave an example of one of those. For instance, topoisomerase is required for the process of cell division. Like, you actually have to have that go in and cut the DNA at certain points, and it's very precise. Like, think about that. This is a molecule. It doesn't have a mind. All right, it's just zipping through, and it's finding specific locations in your genome. cutting out very small portions of it. And it could do that with any part of it, by the way. So it could literally cut you to pieces if it wanted to. I mean, it's a random molecule, right? So the fact that it does this in very specific times and locations, that kind of demands an explanation, but it's never really addressed. And that's just one example. But there's also things like editase. Again, it has to know your entire genome, just so we can go back and repair a small portion of it. It's insanity to think that that sort of system just generated by itself. That's just the last thing I have to say here.
Awesome. Thank you. Immutable, do you have something you want to share?
Sure. Yeah. So I don't think, and regarding Matt's point first, that I made that I was ignoring the existence of repair enzymes or things like that. I mean, it's a different topic than what me and Sal were originally talking about. To respond to the first thing, or maybe to the last point that Sal made, I don't disagree that creationists should be given a chance. I mean, I think the thing that has to occur with creationism at some point is that creationists need to decide on specific definitions and also specific mechanisms which they are willing to accept in terms of how they're going to account for, say, the diversity of life. I'm sure Sal knows this. AIG and AIG, among other organizations, has had like this back and forth attitude on even the number of like how species should be counted when it comes to issues about the arc, whether or not you want to do it at the species level or at the family level or things like that. I'm willing to concede that certain things like this are things that are being worked on. I mean, I think one possible issue that can occur is that there needs to be some kind of like the name of the game has to be made clear, like when you're talking about this, because ultimately, if the particular mechanism being put forth is false, right, it needs to be revised. And if there's no unified mechanism or unified framework for a creationist to operate under, then that definition can't occur. Regarding the other point, though, I think there's a few points I want to make, particularly with the protein evolution issue. My point in raising the point about LUCA was that regardless of what exactly LUCA is capable of, it probably has at least the basic functions of what a cell can do, of at least what a bacterial cell can do. And the point is, bacterial cells can do a lot. And something that I don't think necessarily was necessarily made explicit, and maybe Sal can correct me if I'm wrong, but there's this kind of, my impression is that there's this kind of insinuation or implication that the way in which these mutations occur have to be, in some sense, guided, or in some sense like different, like not a random process that occurs. The problem with that, one, is that if I recall right, in the 1940s, I mean, there were mechanisms. There's a paper concerning bacteria evolving, bacteria adapting to the existence of virophages, and how these specific mutations, and you can make predictions based upon that about if the mutations are random or not, based upon what you see when you culture these cells and do the numbers. But the other point is that, thank you, Dan. Laria and Delbruck, 1942 is the source for that. So this was done in the 1940s. And another major point is that when we're talking about protein evolution, the thing that evolution cares about, right, is that ultimately the organism doesn't die, right? So it could be the case that certain descendants of an organism acquire some mutation that amplifies some process for metabolism by half an order of magnitude, which in that particular niche allows that organism to reproduce quicker and better and more reliably than its than organisms which do not have that particular change in their genome. And so the idea that these kind of mutations need to be directed, I mean, I would need more argument for that, but at the very least, there's some studies I could point to that seem to push back on that notion. And the other is, and that the other reason I make that point is because if the argument is that certain mutations must accumulate in a non-random way to produce certain effects, or to produce some of the variation we see, yet we can find examples where you would expect mutation to behave in a certain way, but it doesn't, right? And by that I mean the mutational pattern to affect a genome in a specific way, but it doesn't. Well, that is an argument against mutations being a kind of directed mechanism rather than something which is stochastic, which is the process that mutations generally understood to occur in.
Awesome. Well, I said I wasn't going to respond, so we'll let Immutable keep his last point. And thank you guys, everybody, for your participation, both in the chat and on here. Like, I loved this conversation. It was super fun, and I learned a lot from it. So thank you guys for coming on. And we actually got, I think it's we've had some points awarded against Young Earth before, but I think tonight we actually got our first 2 points for evolution. So I'll compile that and I'll present it at the next stream. So thank you, Grayson. And thanks, guys, for participating. See you next time. Appreciate you guys.